A very warm good morning, my dear children. Yes, we are back with the lecture number 3 of cell cycle and cell division. Meiosis is the topic of the day. Yes, Bhumi, Anubama, Yamni, a very warm welcome to this session. Hope you all have watched the previous session of mitosis. I haven't noticed these two people, that is Bhumi and Yamni, in the previous record premiering section of mitosis, whereas Anubama, Akil, and Sheikh Sonali was there. Yes, hope you will be able to follow. Yes, Binaya, very warm good morning. Hope you will be able to follow today's session. So, reminding you to do join the Telegram group if anyone new is watching this session. And the link to join the Telegram group is given there in the description box, and that is t.me/neat underscore biopoint. Yes, so hope it's fine. Do hit the like button, share with your friends, and subscribe. Biopoint, stream the world of bio for amazing videos, lecture videos on all the chapters of NEET. Yes, yes, Bhumi, it's fine. Yes, Suja. Yes, yes, everyone. So let's move on with today's session. So this was the schedule for the day. This was the schedule for the day. Oh God, this rank booster was completed. Right, rank booster was completed. Morphology session interrupted due to network issues. Now we have started the topic of meiosis, and at 8 p.m. you will be having Telegram quiz on cell cycle up to mitosis. So do join this too. Okay, only join this quiz 8 p.m. quiz after watching yesterday's lecture. Okay, because mitosis is included there. Okay, so 9 a.m. session interrupted due to network issues. Sorry for that. We don't know how far will this session to go. So let's try our maximum to complete this chapter today itself. The last and final topic of the day, that is meiosis, the last topic of this chapter. Yes. Yeah, I just forgot to say that morphology session just rescheduled at 2:15 p.m. Today 2:15 p.m. you have the rescheduled session of morphology of flowering plants. It is the division of diploid germ. Oh God. Division of diploid germ cells that reduces chromosome number by half, forming haploid daughter cells or gametes. Yes, everyone, yes, I use called Binaya. So let me say, what is this meiosis? That is a cell of two ploidy dividing to form progeny or new daughter cells of ploidy N or 23. This is occurring in case of reproductive organs, that is during the formation of what process of gamete formation. The reproductive organ, that is cells with the ploidy of 2n, divides into new daughter cells or produces gametes with the ploidy of n or haploid, produces haploid gametes. And this haploid gametes, that is a sperm with 23 nucleus, 23 chromosomes, and an ovum with 23 chromosomes, fuse together to form a zygote with 46 chromosomes. So, why this reduction division is occurring? Yes, Devjit, reduction division is occurring in order to conserve the chromosomal number in the next generation. Right? If this was 2n, if the number of the gametes were 2n, ploidy of the two gametes were 2n, you will be getting the total number of zygote is 46 into 2. The double of 46 will be getting as the chromosomal number of zygote, right? That results in the what? Difference in chromosomal number in the different generations. So we need a continuity of species. So that for that continuity of species with each chromosomal number, we are having haploid gametes. We are having haploid gametes that fuse together to form a haploid gametes that fuse together to form a diploid zygote. Yes. So hope it's fine till now. Hope it's fine till now. Okay, fine. So this meiosis occurs during gametogenesis. So what is this gametogenesis? Gametogenesis is the process of gamete formation. Yes, gametogenesis is the formation of gamete formation, and it leads to a haploid phase in the life cycle of sexually reproducing organism. Yes, from the diploid phase, that the normal body cells are of diploid phase, and in case of sexually reproducing organism, when this uh, what happens when this gametogenesis occurs? When this gametogenesis occurs, what happens is the diploid cell gets reduced to, forms a haploid progeny or a daughter cell. Is it clear till now? Is it clear? So after what this haploid, then how this ploidy is restored after fertilization. After fertilization, this ploidy is just restored back to diploid. Yes, everyone. So hope that's perfectly clear till now, so that I may move to the next session. Next slide. It involves two cycle, meiosis 1 and meiosis 2, but only a single cycle of DNA replication. I will explain all these points after reading the entire slide. Okay. It involves pairing of homologous chromosomes and recombination between them. Meiosis 1 begins after replication of parental chromosomes to form identical sister chromatids at S phase. Four haploid cells are formed at the end of meiosis 2. Okay, so let me explain what I have said so far. Yes. So meiosis. We have said mitosis in the previous lecture as a single phase. That is, after mitosis, what happens? Cytokinesis occurs. Right. After mitosis, cytokinesis just occurs. Whereas in case of meiosis, it involves two further stages, meiosis 1 and meiosis 2. So meiosis is a two-step process, that is meiosis 1 and meiosis 2. That is meiosis 1, karyokinesis occurs. Karyokinesis occurs in case of meiosis 1, that is karyokinesis including prophase 1, metaphase 1, Anaphase 1 and telophase 1. All these phases terms we were learned in the previous lecture. These are the different stages in case of karyokinesis of meiosis 1. Prophase 1, metaphase 1, anaphase 1 and telophase 1. So this prophase 1 has 5 sub stages. What are they? The very first one is termed as leptotin. We will learn what all events are happening in this stage. Okay, the very first one is leptotin stage. The second one is psychotin stage. Yes, the third one is pacitin. The fourth one, it is diplotin. And the fifth one, that is diakinesis. These are the 5 substages in case of meiosis. Meiosis prophase 1 of meiosis 1. Then this meiosis 2 is same as mitosis. Meiosis 2 is same as mitosis. It consists of 4 substages. That is prophase 2, metaphase 2, anaphase 2, and telophase 2. metaphase 2, anaphase 2, and telophase 2. And after this meiosis 2, what process occurring? Cytokinesis occurs. Surely you need to remember these stages, and what else you have to remember is what all things are happening in this stage. This meiosis is one of the most important topic for your entrance examination, and it is frequently asked from this chapter. This what all events are happening in this leptotin, psychotin, pacotin, diplotin, and diakinesis for your boards as well as NEET. Yes, everyone. This is the overall what summary of meiosis, that is consisting of two phases, meiosis 1 and meiosis 2. Is meiosis 1 is only of karyokinesis, that is prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1 has 5 substages: leptotin, zygotin, pacotin, diplotin, and diakinesis. And meiosis 2 is same as that of mitosis, consisting of prophase 2, metaphase 2, anaphase 2, and telophase 2, and cytokinesis. Yes, yesterday we have learned that after the process of mitosis, two daughter cells are formed. Yes, after mitosis, two daughter cells are formed. Whereas after the process of meiosis, four daughter cells are formed. So let's see how these four daughter cells are formed. Yes. So this is what we have discussed so far. Stages of meiosis, that includes meiosis 1 and meiosis 2, prophase 1, metaphase 1, anaphase 1, and telophase 1, prophase 2, metaphase 2, anaphase 2, and telophase 2. Yes, only the Abhijit is here. Where are the others? Yes, come on. Yes, Anupama. Yes, Vinaya. Yes, Bhumi. Yamni. Okay, fine. So let me move on with the next session. The next slide, that is prophase 1. We are going to learn in detail what is happening in case of prophase 1. Okay, very focus, because very important. Yes, Sheikh Sonali, very important. So before starting this, let me say something more. So meiosis 1, it is the actual reduction division, that is 2n reducing to n. Whereas meiosis 2, that is same as mitosis. What happens there? Equational division occurs. Equational division, that is the same process occurring in case of mitosis. Yes. So what happens? Karyokinesis followed by cytokinesis in case of meiosis 2. Whereas in case of meiosis 1, karyokinesis followed by interkinesis. Yes, we will learn what this interkinesis and all later. So let's move on with meiosis 1, the very first stage, prophase, which is the typically longer and the most complex, not more, the most complex stage of meiosis 1, that is the prophase. That include 5 points, that is leptotin, zygotin, pacotin, diplotin, and diakinesis. Yes, everyone, these are the 5 substages of prophase 1. Let's learn in detail what all are occurring in prophase 1. What that is the very first stage, leptotin or leptonema. Leptotin or leptonema is the very first stage. So guys, one thing I have to remind you before moving on with this session is that sometimes I will not be, sometimes you will not understand the points that are given there in these slides, because those are some such complicated points in some of the slides. Okay. So what you should do is you should be very focused in what I am dictating, because points are those which I am gonna say from now. Okay, the actual points are those which I am gonna say from now. So if you want me to repeat, you may ask, or else you may note it at the time while I am speaking. Yes. So this is this stage. I'm gonna teach, or I'm gonna explain with the help of this diagram. So this is the stage that is prophase one. This is what is known as prophase one of meiosis one. I mean prophase one. Which stage? Leptotin. Leptotin stage of prophase one. Yes. What is happening in this leptotin stage of prophase one? All of you know that the very first thing that should happen, same as that of mitosis, is that the chromosomes. Yes, the chromosomes should become visible. Chromosomes should become visible. Yes, the second point is that they start to get condensed. Condensation of chromosomes. Condensation of chromosome. And next what happens is that they start to pair each other. They start to pair each other. So all of you know that inner cell, yesterday itself I have said, this may be the chromatin fibers initially. Very sorry. Highly coiled. Yes. So during or at the time of division, they get condensed and form duplicated chromosomes. Yes, they get condensed and form duplicated chromosomes. That is what. And these two chromosomes, as shown in this diagram, they start to move, they start to attract each other. That's a simple term for what? Saying. That is, they come closer. That is what is said as they start to pair. Pairing starts. Yes, are you able to follow? Are you able to follow? Okay, fine. So now let's discuss what are the major events that are going to happen in case of this prophase. So leptotin stage, that is the chromatin fibers become long and slender. Chromosomes, nucleus enlarges, they get condensation of chromosomes, and they start to pair each other. Yes, Ravni. Yes, Bhumi, Anpama, Sheikh Sanali, everyone. Thank you so much for your responses. So that's what happening in case of leptotin stage of prophase. Yes. Next is Psychotin. So let's discuss what is happening in case of Psychotin, the second stage of prophase 1. Yes. So do you know what is its non-sister chromatids? Guys, do you know what is sister chromatids and non-sister chromatids? Do come and fast. Okay, guys. So anyway, let me say it for you. Those who don't know, yes, Vinaya. Surely I will be saying for that. So you can see the two chromosomes that I have drawn here. Please wait. Let me connect the charger to my mobile. Okay, fine. Then only your chat will be visible for me. Okay, fine. So you can see these two chromosomes that I have drawn here. You can see this is known as, yesterday as we have discussed, this is known as sister chromatids. That is the two chromatids from a same chromosome, from a single chromosome. Yes, Sheikh Sonali. Two chromatids from a single chromosome is what is known as homologous chromosome. So if the chromosomes are taken from one from this and from one from other, then what we will say it as non-sister chromatids. Then it is said as non-sister chromatids. So understand clear about the idea of what is sister chromatids and non-sister chromatids. Yes. Yes. Hope it's fine. Yes, dears. So let's move on. So now let's discuss about the stage of Psychotin. What all are the events that are occurring in case of Psychotin? So in case of Psychotin, the very first thing that you have to notice, synapses occurs. What is this synapses? What is this synapses? That is the non-sister chromatids of homologous chromosomes. These chromosomes are known as homologous chromosomes. The chromosomes that are given here with the red and blue colors are known as homologous chromosomes. And the non-sister chromatids, as we have discussed, there is two chromatids from different chromosomes. They just overlap each other. That is, one just come above the other, starts overlapping. As you can see in this figure, the blue chromosome just overlap the red one. Yes. That is what is said as synapses occurring. Synapses of non-sister chromatids that is overlap each other or starts pairing accompanied by the formation of synaptonymal complex. So this process of synapses or this overlapping is done with the help of a complex structure called synaptonymal complex. Guys, if you have any doubt, do comment it. If this prophase one gets over, we could move very fast with metaphase, telophase, anaphase, and all. Till prophase one, it's a great issue. Most important. That's why I'm moving very slowly. So that's about zygote stage. Then another point that you have to discuss, we will learn from here. Chromosome becomes more condensed and similar chromosomes starts pairing together, and that is what is said as synapses with the help of a complex structure called synaptonymal complex. Yes. So what is the synaptal complex? Not synaptal. It is synaptonymal complex. Yes, synaptal complex is a structure that helps in the attachment of these two chromosomes together. We have said that they are overlap each other, right? They are overlap each other. We have said about that. Yes, guys, is the audio breaking? Do comment. Bivalent. Yes, I'm gonna say that, Sheikh Sinari. Do yes, it's too complicated, too much complicated. Yes. Yamni have seen. I will explain that this is made up of. Please wait. I'm just coming to the point. Okay, fine. So it is made up of synaptonymal complex. That is, these are overlapping each other, and that is known as synapses. And that is overlapping is with the help of synaptonymal complex. So what is this made up of? Synaptonymal complex. Yes. YPC. That is synaptonymal complex. Okay, so this synaptonymal complex proteins are there. Proteins named SYPC proteins or synaptonymal complex proteins. There will be SYPC protein 1, SYPCP protein 2, all those things. There's no need of that. So don't need be much complicated about that. Okay, so that complex structure help in the what process of synapses, and that complex structure is known as synaptonymal complex, and this pair of chromosomes is known as homologous chromosomes, and this pair of homologous chromosome after the process of synapses forms a bivalent. What is this bivalent? Please wait, guys. Just highlighting that. So these two chromosomes are just overlapping each other, right? This together forms a single chromosome, and that is known as bivalent. That is two chromosomes each, bivalent, or it is also known as a tetrad. That is 1, 2, 3, and 4. Four of them are just overlapped each other. Therefore, it is also known as tetrad. So, bivalent or tetrad. Each homolog pair of homologous chromosomes is a bivalent or a tetrad. So, hope that's yes. Just we have to study what are the events happening in these, or we should understand how it is going on also. You should understand how what is going on also. That is why we are learning these characteristic events here. So you that is asked for the exams also. So how Psychotin is also clear. Is it fine? So that we may move with Pacitin. And after that you have diplotin, diakinesis, then a lot to complete. Yes, okay, dears. Yes, I have got Devjit, Sheikh Sonali, Binaya, Bhumi, Yamini, Anupama. Where are you guys? Is it fine? Yes, Ravani. So pactin is comparatively a longer phase bivalent chromosome split into similar chromatids. This stage is called as tetrax. Appearance of recombination nodules at which crossing over occurs leads to recombination of genetic materials of the homologous chromosome. Recombination is completed at the end of pactin. These are the points that we are going to cover now in this slide. You can see this is how these chromosomes are just overlapping each other. Yes, that is when such a phase came. Another chromosome will join the same, like a double helical structure of a DNA. Yes, I was asking whether we should just by heart like synapse takes place in which stage, like that. Yes, 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 yes. That are the questions that are asked. Synapse occurs in which stage of prophase one? Options for MCQs, options will be given, and you should answer about that. Yes. So look here, you can see this how they are paired. They all just overlapped each other. They are intertwined, right? They are intertwined. So in these cases, so do notice the points that I'm just highlighting now. Do notice the points which I am going to highlight now. This part, oh, it's not able to see. This part, this part, this portion, this portion, this portion where all these intertouch each other, right? Interlink each other. These portion, certain structures for recombination called recombination nodules appears. Certain structures called recombination. Oh God. So these structures called recombination nodules occur, forms at the sites of crossing over. So what is this crossing over? Crossing over is a process in which the non-sister chromatids exchange their genetic material between each other. Yes, these two chromatids exchange, that is this blue. We consider this as the father's chromosome and consider this as the mother's chromosome. So they will just interlink each other and the characters from mother will be moved on to this red color chromosome and from red color will be moved on to this blue color chromosome. Both will be interlinked each other, just will be exchanged each other, and that process is known as crossing over. That is the process where exchange of genetic material occurs between the non-sister chromatids of homologous chromosomes, and at the site of crossing over certain nodules of recombination called recombination nodules appear. Yes, and the enzyme that help in recombination is recombinase. Recombinase is the enzyme that help in recombination. Yes, bivalent or tetrad is clearly visible at this stage. Yes, guys, are you able to follow? Even it's getting confused. Yes, I could understand that this is one of the most difficult topic of this chapter. Yes, yes, but a little bit complex. Not a little bit, dear. I understand your situation since you are hearing it for the first time. It is not a little bit, it's a highly complex for you. Yes, but don't worry, in the very next day, if we are completing this today, if we are completing this chapter today, surely we will be doing, I will be coming with NCRT in the very next day itself. Don't worry with that. Yes, yes, don't worry, Bhumi, we will do it. Today also, we will, after doing recap only, we will be moving on to next phase. Yes, so that's about Packitine. It's a comparatively longer phase. This stage, tetrads all are clearly visible. Appearance of recombination nodules. It leads to the recombination of genetic material. Crossing overs occurs. So, in which stage of meiosis death or prophase 1 death crossing over occurs? Packitine. That's the answer. Okay. So, recombination is completed by the end of Packitine. That's all about Packitine stage. So, actually, in class 10, we had this chapter, but not that much detailed. Yes, Debjit. Debjit, have you studied ICAC in class 10? For the students of ICAC, they had the second chapter in case of biology, that is the structure of chromosomes and its part. They had, yes, yes, Debjit, yes. Prophase, Metaphase, Anaphase, Telophase, all those things, mitosis and meiosis all you have learned, but you haven't learned about the substages of meiosis, right? Yes, I have Scott. Substages of meiosis is the most difficult and complex topic, and that's what we have learned in this higher class. Yes, so this is the figure or diagram for what Packitine stage that we have discussed now. Crossing over occurring, yes, appearance of recombination nodules, the stage at which tetrads or bivalents are clearly visible. That's all for that's all for pactine stage. Next is diplotin. Next is diplotin. So now everything has happened. Now you can see, if you look here, you can see. Wait. I can't zoom, right? Yes, yes. Yes. If you look here, you can see that the red color of this chromosome just get overlapped or just get passed on to the blue chromosome, and the blue just passed to the red chromosome. That is what occurred after the process of crossing over. Yes, that is the exchange of genetic character or genetic material. Exchange of genetic material occurred. Is it clear? And the X-shaped structure formed. This X-shaped structure formed in the previous case also. You may see in the X-shaped structure formed. You can see here the X-shaped structure formed, and that is known as chiasma or chiasmata. Okay, and what happens here? This chiasmata becomes more visible, and that is the site of chiasmata, that was of X-shaped structure, and that is the site of crossing over. Then what happened? These chromosomes move apart, tend to move apart. For in the very first stage, they moved closer, and now what happened? They are moving apart. Yes, they tend to separate from each other. Yes, the recombined homologous chromosomes, they tend to uh, move apart from each other, and that's all for diplotin stage. It's somewhat simple. Dissolution of synaptonymal complex occurs, that is, they tend to move apart. You can see how it has moved apart. Recombined homologous chromosomes of the bivalent separated from each other except at the site of crossovers. The X-shaped structures is known as chiasmata. Yes, the X-shaped structure is known as chiasmata. In oocytes of some vertebrates, the plotting stage may last for months or years. Not much important. What important is that chiasmate X-shaped structures, then the chromosomes tend to move apart. Dissolution of synaptonymal complex. Bivalents get separated. This is what occurring in diplotin stage. Is it clear? Is it clear? Yes. Give me hearts on the screen if it's fine, so that it could move fast. After this diakinesis, we are going to start with metaphase. Hope we could move bit more fast more than that. Yes, Ravni, I scored. Anpama, Devjit, Sheikh Sonali. Yes, everyone. So Binaya, sir, single X-shaped chromosome or GHTPA. Binaya, can you make it clear? Yes, you may post it. I am just moving on with this diakinesis stage. What happens is terminalization of chiasmata. So we have learned what this chiasmata is, the X-shaped structure. So terminalization occurs. I'm just showing only one chiasmata. You can see this chiasma or chiasmata. moved at the terminal region. Yes, from here, since it is moving depart, it will just move from here to the terminal region. So that is what is meant as terminalization of chiasmata. Fully condensed chromosome. Chromosomes are fully condensed in this stage. Meiotic spindle originates from the poles to prepare the homologous chromosome for separation. Then the another important point is that nucleus and nuclear envelope disappear. So single X-shaped chromosome or both the chromosomes are known as chiasmata. Here, look this chromosome. Look here. This part you can see an X-shaped structure, right? That X-shaped structure is known as chiasmata, and it's not regarding the count of chromosomes. I feel this what you have asked. This X-shaped structure is known as chiasmata and not the number of chromosomes. Okay, fine. Yes, you can see here what's happening. The centrioles just moving apart towards the poles. The terminalization of chiasmata. You can see the chromosomes start to withdraw from each other. Then what else? Chromosomes are fully condensed and mitotic spindle beginning to form. You can see aster is started to radiating. That leading to the formation of mitotic spindle fibers. Yes, and by the end of diakinesis, what happens? Nucleus and nucleolus and nuclear membrane disappears. Yes, exactly. They are actually the points we are crossing over because those are the egg-shaped structures. Yes, so that's all for diakinesis and prophase one. So that we can move with metaphase one. Yes, Metaphase 1, what is happening is spindle formation is completed. You can see the formation of spindle fibers from this case of uh, what centrioles. Chromosome align at the equatorial plane. You can see at the equatorial plane the chromosomes are aligned. The bivalent get arranged at the equatorial plate. Microtubules from the spindle get attached to the pair of homologous chromosomes. Microtubules from the spindle get attached to that is microtubules from the kinetochores, and those kinetochores are the places where the spindle fibers attach. Same as that of mitosis as we have discussed. This is the same events occurring in mitosis also. Metaphase the same as that of mitosis events. The only difference is that the chromosomes are bivalent. Yes, everyone. Microtubules from opposite poles attached to other homologous chromosomes with the help of spindle fibers. Yes. Give me thumbs up if metaphase also is perfectly fine. Yes, Vinaya, Devjit, Shravani, Anupama, everyone. Thank you so much. So next is anaphase. From this anaphase, as we have discussed, they get split up. That is, this bivalent will be moved on to two opposite poles. That is, spindle fiber starts to withdraw. Reduction of the spindle fibers, and this is the phase where this anaphase is the phase where actual reduction occurs. That is, 2n gets reduced to n each, right? 2n gets reduced to n each. So anaphase 1, the homologous chromosomes separate while sister chromatids remain associated at their centromeres. That's all about anaphase 1. Yes, anaphase is also easy, super easy. Yes, prophase is a bit confusing. Don't worry, I will be will be coming with a single session for prophase 1 only. Happy? Happy with that? So that I could move with telophase 1. Telophase, what's happening is that, yes, yes. Only after that we will be doing exams on or telegram quizzes on meiosis. Only after making you perfectly clear with prophase. Okay. So in telophase 1, nuclear membrane and nucleolus reappear. We have said that at the stage of diakinesis, at the end, they get disappears. So they get the reappear in case of telophase 1 and 2 haploid daughter nuclei are formed. You can see 2 haploid daughter nuclei is formed. Right? And that is known as a dyad. Since 2 of them are formed, that is known as a dyad. After this, cytokinesis may or may not occur. After a short interphase, it is followed by meiosis II. So, after meiotic division, meiosis 1, cytokinesis do not occur. Yes, cytokinesis does not occur after meiosis 1. So, then what happens after meiosis 1? It enters a short resting stage that is known as interkinesis. And after that, then starts the meiosis II. We said that after mitotic division, there will be interphase, and after that, again, mitosis continues. Here, meiosis of 2 phases, so meiosis 1, interkinesis, and meiosis 2. Yes, everyone? Is it clear till now? Is it clear till now? DNA replication do not occur in telophase 1. Yes, guys. So we are moving on with Metaphase 2. I mean, Meiosis 2. Very sorry. Meiosis 2. Yes. Oh, God. Guys, are you able to follow? I haven't asked you. Are you able to follow? A very little topic. I think 4 more slides left. 1, 2, 3, 4, and 5. 5. Then the overall roundup and significance. So please tomorrow. Meiosis 2 is same as that of mitosis. Is it, sir? Yes, exactly. Meiosis 2 is same as that of mitosis. So we need to self-study also to master the concept. Yes, dear. If you are just confused with the Prophase 1, just leave it. Surely I will be coming a single session on Prophase 1. Yes, guys. So what's your decision to continue this Meiosis 2 in the next class or to continue it today? Everyone should answer. Shravani, Bhumi, Vinaya, Devjit, Ayoskot, Anupama, everyone. Tomorrow, today, tomorrow, Vinaya, Shravani today, Bhumi tomorrow, Anupama today. Vinaya, where are you? Sheikh Sonali tomorrow, sir. Okay, then it's fine. We will do it tomorrow. Don't worry about that. I'm not giving you overloads for that. Guys, and another thing that I have to remind you is that I will be giving notes of what we have learned in diplotin, psychotin, packaging, and all. Now, soon after this session in the NEET group, not in our BioPoint group, in the NEET group. Yes. So I could be able to provide you with the PDF only after completing Meiosis II. So do cooperate, do watch it from the YouTube. So since all these are did into a single session, we will do it tomorrow itself. Okay. We will once again explain Prophase 1 also tomorrow. So do hit the like button, share with your friends and subscribe. BioPoint, stream the world of bio. That's all for today. It's me signing off from your favorite and my favorite BioPoint, stream the world of bio. Bye-bye.